Well, thank you very much for the invitation to participate in the COE Symposium. It's very much an honor. Let me also start with an apology to the non-immunologists here, because it's always good to have a sleep after lunch, and this is your moment to do so. By way of introduction, I want to show you this painting. I'm sure you've seen it many times. It's a very famous painting, the first clinical case report of clinical transplantation. It's a young man receiving a limb transplant. And the surgeons are the patron saints of modern medicine, Saints Damien and Cosmos. But I show it to you for two reasons: to point out differences from the modern day. The most striking difference is here, because I've had the pleasure and privilege of working with many transplant surgeons in my clinical career, and they're people for whom I have great respect, not least because of the kind of technical achievements we've heard of this morning with living-related liver transplantation. But of all the surgeons I've met, I've never met any surgeons to whom you would award a halo. So that's the first contrast with the modern day. The second is the extraordinary optimism of these surgeons, because you'll notice that the operating room assistant has put two shoes beside the operating table, imagining that this patient is going to walk away on two legs, which, given the insurmountable nature of the challenge of a limb transplant, is highly unlikely. But I want to tell you one or two pieces of data concerning transplantation tolerance which perhaps contribute to possible optimism about achieving this elusive goal at some point in our lifetime. So I would suggest that there are three major problems that limit the clinical applications of transplantation as we currently practice it. The first is the morbidity and mortality associated with protracted immunosuppression. The second is a phenomenon loosely known as chronic rejection, which leads to the ultimate demise of most transplants. For kidney transplants, the half-life is around 10 years. And the third perpetual problem, which actually only gets worse, which is the shortage of organs available for transplantation. And I would go on to suggest that if we could achieve immunological tolerance, this would address all three issues. Firstly, because it would allow drug-free transplant survival, eliminating the first problem. Secondly, as judged from experimental models, it would limit chronic rejection. And because it would extend the longevity of a transplanted organ, it would reduce the number of patients going back onto the waiting list, for example, for kidney transplantation, and thereby address the organ shortage. Now, before getting into any experimental data, let me remind you about the two pathways whereby foreign major histocompatibility complex molecules can be recognized. So the pathway which we're most familiar with is referred to as the direct pathway. It's a very vigorous high-frequency response, indeed uniquely so, and it involves recipient CD4 and CD8 T cells recognizing intact donor class 2 and class 1 peptide MHC complexes largely on the surface of donor cells. This drives a uniquely high frequency of response and causes the mixed lymphocyte reaction in vitro, and probably is the major driver of acute early transplant rejection in vivo. However, to stimulate these kinds of T cells, of course, they need more than simply MHC recognition. They also need co-stimulation. And we first suggested over 2 decades ago that in the absence of co-stimulation positive donor cells, that a second pathway might play a more important role. This is known as the indirect pathway, but in fact describes the way the immune system responds to all conventional protein antigens. So the foreign donor MHC molecules are internalized by a recipient antigen-presenting cell, broken down into peptides, and presented in the context of recipient or self MHC class 2, driving CD4 help for a variety of graft-destructive effector mechanisms. Now, in contrast with the direct pathway, this is a very low-frequency response, as is true for all other peptide antigen responses, but arguably can be clonally expanded in vivo. So I want now to draw one or two lessons from what we've learnt from the rodent models that have been worked on in experimental transplantation systems over 2 or 3 decades, and then draw, again, a couple of lessons from what we've learned from patients. And they both point towards the same kinds of conclusion. So the first lesson from these rodent models I want to draw is that tolerance is much more easily achieved when the MHC incompatibility between donor and recipient is either limited or even absent, so that the response is only against minor antigens. The corollary of that is that tolerance is impeded if T-cell death is prevented. And that's been shown in a variety of systems, but I've highlighted here some experiments from Terry Strom and Larry Turker in the late 90s, where they made animals, or used T-cells from animals that had a survival gene, and it was almost impossible to achieve tolerance under those circumstances. And finally, further work of Terry Strom, some very exciting data, where he shows that tolerance is favored by deliberate deletion. And here he's using an IL-2 fusion protein to drive activation-induced T-cell death of donor-specific T-cells. So based on these kinds of data, and indeed much other supporting data, I think most of us would conclude that in the context of extensive MHC incompatibility, a degree of deletion of that high frequency of direct pathway alloreactive T cells may be required if tolerance is to be the outcome. The second principle that I want, or lesson I want to draw from these rodent models, of course, is very familiar to this audience, because the story goes like this, that if you can achieve tolerance in a rodent recipient to an MHC incompatible graft, very often, in many of these models, you can transfer that tolerance, almost always with CD4 T cells, into a naive animal, and the transferred T cells will serve to protect a challenge graft from the same strain as that to which the T cell donor animal was tolerant. So this is antigen-specific active tolerance mediated by cells that you can call suppressor cells or regulatory cells. And this was discovered long before the elegant work that Shimon has just presented was ever understood. So the whole field of regulatory cells has now exploded. And I'm not going to talk through all the various kinds of regulatory cell that have been described, but I am going to focus on this population here. And let me pay credit to Shimon Sakaguchi, because really he has fathered this field and restored the honor of suppressor cells, and in doing so renamed them regulatory cells. But few people's work has had a bigger impact on modern cellular immunology in our generation. So the second lesson from rodent models is that tolerance is maintained by dedicated regulatory cells. Now, what about patients? What have they taught us? Well, we and a number of other groups have done a lot of immune monitoring of transplant recipients, and that's highlighted conclusions that I think are related in some way to what I've just said about rodents. So this is a study published a few years ago where we looked at 22 recipients of haplotype mismatched living-related kidney transplant recipients. And we looked at their frequency of IL-2 secreting CD4 T cells using a limiting dilution assay. The frequency is shown on a reciprocal log scale, so very high frequencies up here, really unmeasurable frequencies down here. And in each patient, we measured the frequency against the donor, shown in blue, and equally mismatched third parties, shown in red. And these patients were studied, I should say, some years after transplantation. But what you can see is that in all patients, the frequency against the donor was significantly lower than that against third party, sometimes subtly so, very often strikingly so, and a number of these frequencies really are falling into the unmeasurable range. But you'll see also that this applied equally to a cohort of patients who had the classical features of chronic allograft nephropathy or chronic rejection, suggesting that this direct allo response had little, if anything, to do with the pathogenesis of that ongoing graft decline. Now, in contrast, if we studied this cohort of patients and contrasted them with this cohort of patients and used a limiting dilution assay only designed to measure the indirect response, the picture was quite different, and the chronic allograft nephropathy patients had a clearly higher frequency of indirect pathway T cells against the donor compared to those with good and stable graft function. So all of this would point towards the indirect allo response being the major ongoing threat to long-term transplant survival. And so to summarize what I've just shown you in very diagrammatic form, it appears that this is the case, that the direct pathway allo response starts out, as I mentioned, as a very high frequency, vigorous response that tends to decline with time in most patients, arguably due to an ongoing encounter with co-stimulation deficient donor cells. There are other interpretations. The indirect pathway that starts out as a very low frequency, wimpy response may be chronically expanded by the persistent traffic of recipient dendritic cells through a partially inflamed tissue, taking those antigens to the lymph node and presenting them in peptide form, causing grumbling clonal expansion. Now, returning to these rodent models for a moment longer, as I mentioned to you, tolerance can very often be transferred with CD4 T cells. Several groups have tried to address the specificity of the T cells that can transfer tolerance and have concluded that those T cells themselves, the regulatory or suppressor cells, have indirect allospecificity in support of this notion that the response that most needs to be controlled in a sustained manner is that indirect response. So all of that is the basis for the focus in my own research group in finding ways to effectively regulate this indirect response as a way of sustaining long-term transplant tolerance. So I'm going to describe two approaches, one very briefly and one in a little bit more detail. Preclinical approaches or preclinical data supporting strategies to promote transplant tolerance. And they're both focused on this ambition to generate indirect pathway regulation. So the first approach is an obvious one, and that is to try to induce regulatory cells of this specificity in vivo by some form of negative vaccination. In other words, to induce antigen in a form that will turn the immune response off rather than turn it on, and again with a focus on the indirect response. And the cell that we chose to use, not surprisingly, is the dendritic cell, because as I'm sure you know, while this can be the most exciting T cell, exciting antigen-presenting cell a T cell can hope to meet, it can also be modified to become a cell that promotes tolerance and regulation. So we used a rat model of kidney transplantation, which in fact was where my immunology career began, using two rat strains, August and Lewis, and we chose as our tolerance-inducing cell an F1 Lewis by August dendritic cell. And the logic here was that we felt this cell would be most efficiently presenting the August alloantigens as peptides indirectly with Lewis MHC, because we know that through constitutive antigen processing, that is exactly what happens, that MHC peptides from one molecule are presented in the context of another. So by taking this F1 cell that was constitutively engaged in indirect pathway presentation, modifying it to render it tolerance promoting, and we did that with dexamethasone, and using that as a negative vaccine, we would see whether or not we could prolong the survival of an August kidney allograft. And the answer was yes, in combination with other interventions. So the first thing to say is that when we gave the modified dendritic cells, we gave a small dose of CTLA-4 Ig to inhibit representation by recipient dendritic cells. That was a week before transplantation. And then we gave 2 weeks of cyclosporin and then withdrew the drugs. And so you can see the animals that went through that protocol all enjoyed indefinite survival, and even at 100 days had very good graft function and at sacrifice good histology. All the controls rejected. And the most important one I draw your attention to is if we used fully allogeneic August dendritic cells, treated in the same way with the same combination of other interventions, there was no prolongation, and also that this effect had specificity because brown Norway graft survival was not prolonged. So we think this is an approach to targeting the indirect pathway in vivo. But I want now to turn to the alternative, which is that Shimon Sakaguchi just referred to, of adoptive therapy with customized regulatory cells selected and expanded ex vivo. So the first set of studies will address the following questions. Firstly, are cells with allospecificity for donor antigens superior to polyclonal autoreactive cells? And then in the second set of data I'm going to show you, are cells with direct or indirect allospecificity the more effective? So the first study is a published set of data, published I think late last year or early this year, where we simply purified CD4, CD25 population in a conventional way from mouse lymphoid tissue, and then set up cell lines in vitro by repetitive stimulation with autologous dendritic cells pulsed with a peptide of a class 1 alloantigen, in this case K of B, the responder being CBA. At the same time, the postdoc who did this, Della Golshayan, set up lines with CD4, CD25 negative cells for comparison. So the first observation was that these cell lines are remarkably robust and stable during in vitro culture, and Della was able to maintain these lines for months with this repetitive dendritic cell-based stimulation. They also remained faithful to their original phenotype because the lines derived from 25 positive cells are shown in blue. The lines derived from the starting population of 25 negative cells shown in red. This is a week after their last antigen stimulation. Of course, the CD25 negative line becomes 25 positive because this is an activation antigen, but the 25 positive line is always much higher for CD25 expression. They also expressed high levels of intracellular CTLA4, high levels of GITR, and importantly, for several months, retained the ligands that would enable them to traffic appropriately in vivo, namely CD62 ligand and CCR7. Not surprisingly, they were very strongly expressing FOXP3. This is at the start of the cultures. This is after 2 months in culture. Still, the line derived from 25 positive cells, much more highly FOXP3 positive than the line derived from 25 negative cells. Even more importantly, these cells become more potent in their suppressive function. So this is a typical mixing experiment where you're stimulating 25 negative CD4 cells with anti-CD3 and autologous antigen-presenting cells, and you titrate into the culture increasing numbers of freshly isolated CD4, CD25 cells. You really only see impressive suppression at a ratio of 1 to 1, but the cell lines show 90% suppression at a ratio of 1 to 8, so more potent in their suppressive function. Did they have any bigger pardon, before turning to that, what about their trafficking properties? Well, we did short-term and long-term trafficking studies, short-term with CFSE, and long-term by transducing the 25 positive cell line with a retrovirus encoding GFP, and then put those into an animal carrying an antigen-positive skin graft. And 40 days later, and indeed Della then went on to look at 80 days later and saw the same thing, you find the GFP-positive cells revealed on the vertical axis in the draining lymph node and in the graft itself, but very few and irrelevant lymph nodes. So these cells survive long-term in vivo after protracted in vitro culture, and they traffic to and accumulate at sites where antigen is available, both in the lymph node and in the transplant itself. So to assess their ability to prolong graft survival, we used one of these heavily manipulated mouse models where essentially you render a mouse T-cell deficient and then put back effector cells alone, regulatory cells alone, or a combination of the two. This is a skin graft model putting an antigen-positive skin graft onto the CVA recipients. And you can see that the grafts are all rejected within 25 or 30 days if only the effector cells are put back. None of the animals given the regulatory cells reject, and about 70% of the animals show prolonged graft survival with the mixture, and these grafts, I have to say, look beautiful with good hair growth, and the grafts survive indefinitely. Was there any specificity in this effect? Well, the answer appears to be yes, because if the animals were grafted with a third-party graft that, again, only has a single class 1 mismatch, but a different class 1 mismatch, plus minor antigens, there was no benefit in terms of graft survival. Now, having said all of that and shown that there is evidence for specificity, I'm pretty confident that the majority of the cells in those cell lines were autoreactive and not allospecific. And for that reason, we looked at an alternative possibility to invoke specificity, and that is to confer a specificity on these cells by T-cell receptor gene transduction. So again, focusing on the indirect pathway, a Chinese postdoc, Julia Tsang, cloned the T-cell receptor genes from a T-cell receptor transgenic that had been made by Pat Busey, and this transgenic is specific for a peptide of K of D presented by the class II molecule A of B. So she cloned the cDNAs and put them into retroviral constructs and then transfected or transduced 25 positive cell populations. She showed in short-term flow cytometric analysis that indeed the transduction was successful, but it was difficult to sustain those transduced cells long-term in vitro. So for that reason, she turned to establishing cell lines of regulatory cells and then doing the transduction. The first cell lines she made were simply autoreactive, so she stimulated them with autologous dendritic cells and transduced then with the alpha and beta chain cDNAs, and you can see a week later that you've got a population of double positive cells expressing the relevant alpha and beta chain transduced or encoded by the transduced genes, but she couldn't sustain or expand the cells that were transduced by in vitro stimulation. So what she did turned out to be an intelligent and successful move. She made an alloreactive line, a line with direct allospecificity against BALB-C, that's H2D stimulators, by repetitive stimulation with BALB-C dendritic cells, then transduced with the indirect pathway receptor genes. And you can see then that she was able progressively to expand the number of receptor-positive cells, again by stimulation with autologous dendritic cells pulsed with the KD peptide. And indeed, after 3 months of in vitro restimulation, she got cells that were practically 100% positive. But that difficulty with the autoreactive cells illustrates what I think is an interesting challenge if you want to make antigen-specific regulatory cells, which I've tried to illustrate here. So if this is an autologous antigen-presenting cell with MHC molecules occupied with a whole variety of autologous peptides, and this is a polyclonal population of regulatory cells, then they will respond to the autologous dendritic cell as indeed you would expect they would. If you then pulse your dendritic cell with an exogenous antigen peptide, such as the ones I've been describing, and you ask the regulatory cells to respond to this, you'll tend to find that the response to this, which would require a cross-reaction, will get drowned out by the response to the autologous ligands. And that's a challenge, I think, that we need to overcome, and I've shown you one way to do that. So these retrovirally transduced cells, by rights, should have 2 specificities, because they were initially expanded against BALB-C and then transduced with this receptor with indirect specificity for K of D. So we then went on to test that by taking the line that was generated simply against BALB-C. This is the regulatory cells repetitively stimulated with BALB-C. So this line should have direct anti-BALB-C specificity. And here's the same line transduced with the receptor with indirect specificity for K of D. You're looking at proliferation here as recorded by CFSE dilution. And if you stimulate the 2 lines with autologous dendritic cells with no added peptide, you see a little bit of proliferation, but not much. If you stimulate with the same autologous dendritic cells pulsed with the KD peptide, there's no proliferation by the line that has direct specificity, but very nice proliferation by the cells into which you've introduced the indirect specificity receptor. If you stimulate the lines with BALB-C dendritic cells, then you see very nice proliferation by the line that was already selected to have direct specificity, but also by the cells that you've transduced because they have 2 specificities, and a low-level allo response to CBA, a third-party set of alloantigens. So then Julia went on to test the ability of these cells to suppress through the receptor that we had introduced. And this is probably the most complicated experiment I'm going to show. And again, further apologies to the non-immunologists, but this is an experiment to reveal link suppression. So what you did here was took autologous BLAK6 dendritic cells and pulsed them with a peptide of ovalbumin that is recognized by another T-cell receptor transgenic population called OT2. So that's the readout response, and here it is as proliferation. And then she either did or did not add the KD peptide that should be recognized by the transduced regulatory cells to look at whether or not the suppression was enhanced by the presence of this peptide. So here's the mixing experiment at a ratio of 1 to 1. You can see that when you add the KD peptide at 2 different concentrations, you markedly increase the suppression, and the same at a ratio of 3 to 1. So this is peptide-dependent suppression of an irrelevant response, a conventional demonstration of link suppression, and confirms that the conferred retrovirally transduced receptor was indeed active. So then Julia went on to do in vivo trafficking experiments, and what she did was put the transduced cells into animals carrying a KD skin graft. This was, she injected the cells 2 days after skin grafting, and then looked at day 12 in the draining lymph node, irrelevant node, and spleen. She also looked in animals that had an irrelevant third-party skin graft. And what I hope you can see in the draining lymph node, very clear evidence of division. This is CFSE dilution again of the transduced cells, much less so in irrelevant lymph nodes and also in the spleen. And again, very little division in the animals carrying a CBA graft. So again, the cells going to and dividing at the site where antigen's available. So in terms of the effect on graft survival, she first simply put them into an intact, unmanipulated T-cell replete black 6 animal given a B6KD skin graft, and compared either no cells at all, or the cells with direct anti-Balb-C specificity, or the cells that have been transduced and therefore had indirect as well as direct specificity. And the only prolongation was by the cells that had the indirect pathway receptor. That's not surprising because this is a class 1 antigen, and you wouldn't expect the direct pathway CD4 cells to recognize this graft. So then we looked to see whether or not we could combine this adoptive therapy with a form of depletion, and chose anti-CD8 antibody depletion as a short-term measure. So this would lead to CD8 depletion for a matter of 2 or 3 weeks after transplantation. And we used heart grafts from a B6D2 strain, so this is an F1 strain, B6H2B by D2H2D. And you can see that the control animals that only had CD8 all rejected by around day 25, the animals that had the direct pathway regulatory cells, there was a minimal effect. But the animals that had the cells that had the indirect pathway receptor showed markedly prolonged survival, and these animals are still going at 150 days. Animals that have been sacrificed have reasonable cardiac architecture. So finally, before completing this part of the talk, we've begun to look at the possibility of doing the same kind of thing in man. And I should say that it's considerably more difficult. But these data show you that it is possible to enrich for indirect pathway regulatory cells from human CD4, CD25 high cells. And we're revealing the specificity here by using a class 2 peptide tetramer. So this was work by Shuiping Zhang, a very talented Chinese postdoc. And he stimulated these lines from a DR1 individual with autologous dendritic cells pulsed with a peptide of HLA-A2. And you could see that over time was able to enrich for tetramer positive cells. So this is a tetramer made up of DR1 occupied with the A2 peptide. And indeed, if you sort these cells using the tetramer, you can get very nice enrichment of these cells upon reanalysis. The problem is that they're very difficult to expand into the sorts of numbers that you'd wish to use for any kind of clinical therapy. So that's a problem that we have not yet solved, but seems to me, in theory at least, that it's a problem that should be tractable if we could find the right way to do so. And those cells, I should say, suppress as you'd expect in vitro. So, first set of conclusions, that regulatory cells with indirect anti-donor specificity can be generated ex vivo by repeated stimulation with cognate peptide. Indirect specificity can be conferred on regulatory cells by gene transfer. Those cells traffic to the draining lymph node and to the allograft itself following intravenous injection. That adoptive therapy with such cells prolongs allograft survival. And that cells with indirect specificity appear to be superior to those just with direct specificity, and combining adoptive therapy with such cells with short-term immunosuppression, for example, a deletional strategy, induces long-term graft survival. Now, in the last few minutes, let me turn to a related but a different topic, which concerns the elusive molecular basis of action of this cell population. So, to initiate these studies, we were working with human cell populations that were separated into CD4, CD25 negative or low, and CD4, CD25 high fractions. And we subjected these then to conventional 2D proteomic analysis, followed by mass spec and to gene expression analysis. And out of that came the following findings. So in comparing gels, there was a consistent difference between the 25 negative and 25 positive cells, which I'm showing in magnified form here. So this spot much more exaggerated in the 25 positive cells than 25 negative cells. And so this was picked and analyzed by mass spec and proved to be one of a family of lectins called galectin-1. So then by Western blotting, you'll see that galectin-1 is markedly overexpressed in the 25 positive fraction at rest and after activation, most so at 24 hours and falls off at 48 hours when compared to the 25 negative cells. It's not absent in 25 negative cells, but much less expressed. So, as I mentioned, galectin-1 is a member of a large family of animal lectins. It's expressed by multiple cell types, including macrophages, B cells, some CD8 T cells, thymic epithelium, and some cancer cells, which appears to be an escape mechanism for some cancers. It is largely intracellular, but can be secreted following cellular activation, and either binds back to the cell surface of the cell that makes it or to extracellular matrix. Functions as a homodimer, can induce apoptosis in some cells when added to cell cultures, and it's postulated to have a role in immune cell homeostasis. However, it's worth pointing out that the galectin-1 knockout mouse is grossly normal, albeit recent findings have described the fact that they have exaggerated Th1 responses and more severe, substantially more severe EAE following injection of, I think it was the MOG peptide. Also interesting to note that in the first gene expression study that was done by Ethan Shivak, or reported in year 2000, it was one of the transcripts that they noted to be overexpressed in mouse CD25 positive CD4 cells. So by confocal analysis, you're looking at CD25 negative cells either at rest or after activation in the top panels, and 25 positive cells again at rest or after activation in the bottom panels. You can see there's very little staining in the 25 negative cells, but very strong staining in a significant fraction, around 30% of these cells at rest, and that increases following activation. And at higher power, you can see this punctate intracellular staining. Furthermore, the level of galectin expression correlates with the level of CD25 expression. This is again in human cells, where you see this smear. And as you know, the CD25 high fraction are the fraction that appear to contain suppressive properties. So if you then sort negative, dim, and high, you can see that the level of galectin expression correlates with CD25 expression and is by far most highly expressed in the CD25 fraction. And furthermore, galectin does appear to be related to function in the sense that if you do, again, a mixing experiment and you add in an anti-galectin antibody, you reverse suppression, at least partially, by the addition of the antibody. And that's also revealed in a CFSE system. Here's the control response. Here's the effect of adding a mixture of 25 positive cells. And again, the significant reversal with the antibody. And finally, we've begun to look at the T cells from the galectin-1 knockout mice and look at their properties. The first thing to say is that they have equal numbers of CD4, CD25 cells, which again express FOXP3. But in titrations of their efficacy in response to a polyclonal stimulus, you can see that they are less potently suppressive than their wild-type counterparts. We've now got hold of galectin-1 knockout mice on a black 6 background for a number of in vivo studies, which we eagerly await. So my final conclusion slide, galectin-1 is markedly overexpressed in both mouse and human 25-positive regulatory cells. The levels correlate with levels of CD25 expression in human cells. The antibody against galectin-1 significantly, albeit partially, reverses suppression mediated by human, and indeed now we have data on mouse cells. The cells from the galectin-1 knockout mice are less potent than their wild-type counterparts. And based on this, we would suggest that galectin is an effector of suppression mediated by this cell population. It's clearly not the entire explanation, but we think it is a contributory molecule and look forward to generating further data in regards to that. Thank you very much for your attention. And before closing, let me just draw your attention to the people whose work I've described. So Della Golshayan did the first series of experiments with the mouse lines. The retroviral transduction work was done by Juliet Sang, the human studies by Shui Ping, and Giovanna has been a lifelong collaborator in all this work. Thank you very much.